Next one is the functional groups characteristic of sugar are. So we have to determine what are the functional group that is present in sugars. And sugar, we mean the simplest sugar, that is the glucose, because all other sugars are formed from the glucose. So glucose is the monomer. So if we see the structure of the simple sugar, that is glucose, we can be able to answer this question. So it is clear to us that this is 1, 2, 3, 4, 5, 6. This is a functional group, C double bond OH, and this OH, these are another functional groups. So two functional groups that are present in a sugar molecule is an aldehyde group and an alcohol group. And this aldehyde group contains C double bond O and H. This group is called the carbonyl group. And when a functional group is present and alcohol is present, we call this OH molecule to be hydroxyl. So in presence of this aldehyde group, the alcohol group name is hydroxyl. So out of these options, carbonyl phosphate, no. Carbonyl is present, not phosphate. Carbonyl methyl, no. So these two are wrong. Carbonyl and hydroxyl. So D is our correct option. Now the next one is the Golgi complex participates in. So there are several events that are being given in the statements, and we have to choose one statement in which the Golgi apparatus participates. Now, before going through all the statements, we should know which event take place and in which organelle. So, the nucleus, which is an important organelle of the cell, that participates in the formation of RNAs, which will help in the formation of protein. So, the mRNA that is formed inside the nucleus and that comes out of the nucleus and then goes and joins with the ribosomal RNA that is present on either in the nuclear cytoplasm or in the endoplasmic reticulum. So that helps to form the protein. Now in the rough endoplasmic reticulum, when the protein is formed, then it passes through the several stages of protein modification like co-translational or post-translational modification and leading to the formation of a protein. A proper protein is formed after undergoing modification and folding and everything. A proper protein is formed, and then it goes to the next organelle that is present after ER, and that is Golgi, where the protein sorting is done. Which protein will go where, which protein is destined to go where, that is done by the Golgi apparatus. And now in the mitochondria, cellular respiration takes place as well as in the cytoplasm, because the respiration starts from glycolysis and glycolysis occurs in the cytoplasm. Next, it goes to the TCA cycle. TCA cycle occurs in the mitochondria because the enzymes of TCA cycle are present in the matrix of mitochondria. Then it enters into the electron transport chain and oxidative phosphorylation, which occurs in the inner mitochondrial membrane because the enzymes are present there. Now, the formation of a protein or the amino acid that is taking place in the endoplasmic reticulum, in the rough endoplasmic reticulum, or sometimes in the cytoplasm. And what else? Fatty acid. For fatty acid synthesis and breakdown, that is oxidation, they take place in two different locations in the cell. And their processes are completely different because the substrate that they use to prepare or synthesize a fatty acid and then to oxidize a fatty acid, both are different. Because fatty acid synthesis takes place in the cytoplasm, in the cytosol. But the beta oxidation or the breakdown of fatty acid that takes place in the mitochondria. Now let's check all the statements. Respiration in bacteria, no, because respiration takes place in the cytoplasm and that goes on into the mitochondria. Formation of secretory vesicle, I'm coming to that. Fatty acid breakdown, as I have already said, that detoxification or fatty acid breakdown occurs in the mitochondria. Activation of amino acid, that means the formation of any proteins or activation of amino acid by tRNA, which is coming and joining with the mRNA and rRNA to form the protein, that is all taking place in the cytosol. Now, but for the second statement, the formation of vesicles, what is happening is that the nucleus that is present, it is preparing the mRNA. This mRNA is coming out through the pores of the nucleus and it is joining with the rough endoplasmic reticulum. Suppose there is ribosomes that are present. The portion of the ER which contains ribosome, that is called the rough endoplasmic reticulum. Now, the mRNA transcript is coming here and forming the protein. Protein is undergoing modification through the different phases of this ER and finally it is joining the Golgi apparatus. Now this Golgi apparatus, the function of the Golgi apparatus and this ER is somewhat related to each other because the modification of protein like glycosylation, the post-translational modification or co-translational modification, everything is taking place inside the ER and ultimately it is joining the Golgi for the sorting of protein, like which protein are destined to go to which places of the cell, that is done by Golgi apparatus. So the function of Golgi is to sort the protein, to give the tag to the protein that which protein will go in which organelle, whether to plasma membrane, whether to ER, whether to nucleus or sometime to other cell. Or it can set the protein by forming small vesicle that comes out from the Golgi. So Golgi, while transporting this protein, it forms vesicle and those vesicles are called secretory vesicles. And that is how it helps to transport the protein. So mainly Golgi complex is participates to form the secretory vesicles. And one of that secretory vesicle is the lysosomes, which comes out from the Golgi and that contains the digestive enzyme in the acidic pH medium. Okay. So the answer for this question is the second one.